The purpose of this video is to explain why enzyme are such good catalysts and how does an enzyme can decrease the activation energy. There are in fact three different reasons, or main reasons, explaining why enzymes are great catalysts. The first reason is the formation between substrate and enzyme of transient covalent bonds. These will activate the substrate. It will lower the activation energy. And these transient covalent bonds are formed between chemical groups on the substrate and chemical groups carried by amino acid on the enzyme in a site that is called the active site, where catalysis takes place. The second reason is the formation of weak non-covalent bonds. The formation of each of these bonds results in a release of a small amount of energy, and the summation of this small amount of energy form what is called the binding energy. And the binding energy that is released is the main source of energy that is responsible for the decrease of the activation energy. On a reaction coordinate diagram, the binding energy is represented by the difference in free energy between the transition state in the uncatalyzed and the catalyzed reaction. Now we can determine the impact of the binding energy to the decrease of activation energy. Usually, a decrease of the activation energy by 5.7 kJ per mole results in the increase of the rate of the enzyme by tenfold. Now, most of the enzymes will decrease the activation energy by 60 to 100 kJ per mole. The energy that is released by one weak non-covalent bond is in the range of 4 to 30 kJ per mole, which means that you need only few of these weak non-covalent bonds to be formed for the catalysis to proceed. And that is an extremely important point. The third reason why enzyme is such a great catalyst is the exquisite specificity of enzymes. We can take, for example, beta-galactosidase. It's an enzyme that catalyzes the hydrolysis of a family of molecules called beta-galactosides. A beta-galactoside is composed of a galactose unit linked to an R group that can have different chemical structure. And the linkage between galactose and R group is a very specific bond called a beta-glycosidic bond. And that is the bond that is cleaved in a reaction that is catalyzed by beta-galactosidase to produce galactose and an R group. So beta-galactosidase will function regardless of the identity or the chemical structure of the R group. That is not where the specificity lies. In fact, you can show the specificity by using an isomer of a beta-galactoside. That is called an alpha-galactoside. In an alpha-galactoside, the only difference is that instead of having a beta-glycosidic bond, you have an alpha-glycosidic bond. So there is a slight change in the configuration of the molecule, of the substrate. This small change prevents the enzyme beta-galactosidase to hydrolyze alpha-galactoside. In 1890, Emil Fischer proposed a model that explained enzyme specificity. He assumed that a substrate binds to the enzyme like a key in a lock. That model can indeed explain specificity, but cannot explain the efficiency of catalysis. Let's take a simple model. We have a substrate made of two different groups linked by a covalent bond. This substrate will have to reach a transition state where the bond will be bent and then the bond will be cleaved to release the two products that correspond to the original two domains of the substrate. If we look at the reaction coordinate diagram, we see that the conversion of the substrate into a product needs to overcome a very high energy barrier that makes the rate of the reaction negligible. Now let's take the same example using the Fischer model. So in the Fischer model, the enzyme and the substrate fit tightly together, which means that the substrate cannot reach transition state. The enzyme substrate is extremely stable. It is so stable that its free energy is lower than the free energy of the initial substrate, which means that the energy barrier that has to be overcome is even higher than in the absence of the catalyst. In other words, the enzyme, according to the Fischer model, will not help the catalysis at all. Now, if we take a different model where the tight fit is not between the enzyme and its substrate, but between the enzyme and the transition state, then the substrate will bind to the enzyme. You will have formation of the transition state, so the substrate will be destabilized. You have weak binding interaction between the transition state and the enzyme, and that binding energy will offset the activation energy, and the reaction will proceed at an accelerated rate. You have also experimental observation that disprove the Fischer model. We have molecules that are called structural analogs. These are molecules that have a chemical structure very similar to the substrate, but fail to react with the enzyme. If we take a structural analog that is slightly smaller than the substrate, according to the Fischer's model, this structural analog can enter the active site of the enzyme because it's smaller than the lock, and then this structural analog will react. What we observe is, in fact, that the structural analog do not react. If we take now a structural analog that is slightly bigger than the substrate, what is predicted from the Fischer's model is that the substrate cannot bind, the structural analog cannot bind to the enzyme because the structural analog is too big to fit in the lock. And of course, there is no reaction. What we observe is that indeed the structural analog failed to react, but in some cases we can demonstrate a tight binding of the structural analog to the active site of the enzyme, which is in disagreement with the Fischer model. So in 1958, Daniel Koshland came with a different model called the induced fit hypothesis. Well, this model accounts for the deficiency of the Fischer model and is based on two assumptions. The first assumption is that you have optimum weak interaction between the enzyme and the transition state. And the second assumption is that upon binding of the substrate to the enzyme changes conformation, and that is the induced fit that optimize the position of the reacting group and favor enzyme catalysis. So now if we go back to our example with the structural analog, we can understand why a structural analog that is bigger than the substrate can fail to react with the enzyme but bind to the active site of the enzyme. When we mix the structural analog with the enzyme, it can bind because the active site of the enzyme is in this open conformation. And then once the structural analog is bound, you have a change of conformation, but this change of conformation is not complete because the structural analog hinder the motion of the different region of the enzyme and prevent the correct alignment of reacting group and thus prevent catalysis. In this pi mold simulation, what you can see is the change of conformation of an enzyme called citrate synthase upon binding of its first substrate, oxaloacetate.
Why is there an energy barrier that limits the reaction rate?
So I guess that you didn't fully pay attention to the previous video, where we talked about the fact that energy is required for several transformations that are needed for the reaction to proceed. And these were rearrangement of bonds, formation of charge inside the active site of the enzyme, distortion of the reacting groups that are needed, and so on. So now we know a lot about catalysis and how an enzyme can activate or can increase the reaction rate. We have seen that we have weak non-covalent interaction that contribute to a decrease of the activation energy as well as the specificity of the enzyme. We have transient covalent bonds that are formed that decrease the entropy of the system and consequently favor enzyme catalysis. And then we have this transient covalent bond also contribute to the correct positioning of reacting groups. Now there is another aspect that we didn't discuss yet, which is dissolvation. In the cell, a substrate is surrounded by molecules of water, and there is formation of weak interaction between the water molecules and the substrate. Dissolvation is in fact a process by which these weak interaction between water and substrate are replaced by weak interaction between the substrate and the enzyme. And this weak interaction will contribute to the binding energy and will facilitate catalysis. So in conclusion, catalysis is driven by structure and conformational changes.